Greetings to everyone tuning in, and welcome to this presentation on the Golgi apparatus. The Golgi apparatus is one of many candidates running for the position of presidency this year for the cell because the nucleus is stepping down from its long-time position. We will explain to you why the Golgi apparatus deserves your vote. Golgi for gold. A little bit of background on the Golgi apparatus. It is found in all eukaryotic cells. It is more numerous but also smaller in size in plant cells. It is located close to the endoplasmic reticulum because it works closely with the ER and ribosomes to package their products and secrete them throughout the cell and outside of the cell. As mentioned, it is a protein secreting organelle, meaning it finishes, sorts, tags, and ships proteins produced by ribosomes and within the ER to other parts of the cells as well as outside the cells. But also helps alter carbohydrates attached to the proteins, which are called glycoproteins, and the membrane phospholipids that will be used in the cellular membrane to protect the cell. The Golgi apparatus also creates lysosomes that help clean up the cell and keep it beautiful. A little bit of the structure of the Golgi apparatus. It has two sides, the cis face and the trans face. Vesicles enter through the cis face, where they will be packaged, and as they move along towards the trans face, they will be secreted, and that's where they are finished up. And the cisternal maturation model describes how the vesicles and the products within the vesicles enter the cis face into one of the membranes, as you can see, and then the whole membrane, or cisternae, travels to the trans face, where the finished products are then secreted. And the Golgi apparatus is an example of compartmentalization, where it has a specialized function with many folded membranes to increase surface area. You may ask, how will life function without the Golgi apparatus? It's simple. It wouldn't. Proteins would not be sent to the right locations. They would still be produced by ribosomes, but yet they float around freely within the cell and they wouldn't be shipped to their destination. Secretory cells would not function properly, such as those in the endocrine system, which rely on secreting their products to other cells in order to have the body or an organism function properly. There would be no foundation to the cytoskeleton. The cytoskeleton is basically a railroad system throughout the cell that connects organelles and helps deliver their needed biochemicals. And since the Golgi apparatus is the shipping station for all these products, without it, the cytoskeleton would have no foundation or central station to exist out of. And the Golgi apparatus is in charge of communication and movement of biochemicals outside the cell. So without it, a cell would have to function all by itself. It wouldn't be able to communicate with other cells or send its products. And cells, as we know, cannot do that because cells are specialized and differentiated and cannot produce everything on their own. And if we win, we promise to cooperate with many other organelles, including the rough endoplasmic reticulum and ribosomes. We will continue to receive, make edits, and transport their products for them to remove the hassle. With lysosomes, we will continue to create them to keep the cell beautiful to help clean up the cell. The cell membrane will continue to create products for them and keep them in check and continue to send them nutrients that need to be transported throughout outside of the cell. And lastly, vesicles and vacuoles will continue to create them, and we continue to keep supplying them jobs and promise many more to come. We will make the cell great again. A little bit about other organelles and why they do not deserve the presidency. The vacuole, in animal cells, it basically just holds trash, and it only has two functions at most, while the Golgi body has many. Some malfunctions of the vacuole include Danon disease, which is weakening of the heart and skeletal muscles, and mental disabilities. We don't want this organelle in charge if it can cause all this destruction if it goes wrong. The cell membrane, as the Golgi apparatus does its job on its own, it's 7 jobs. The cell membrane does not. It requires proteins and transporters to move products through it. It can't even function on its own. So how do we expect it to function as president of the cell? It needs help from many other organelles, which therefore, why does it deserve the presidency? You're right. It doesn't. It even can cause Alzheimer's disease. We don't want that to happen. The Golgi body also creates lysosomes. So without us, the lysosomes wouldn't even exist. So how can they be president? If we deserve president and they can be our vice president, but I don't even know about that. And they can cause Tay-Sachs disease if they have malfunctions. The mitochondria, they don't even exist within all eukaryotic cells. And our cells didn't even used to have mitochondria, so why do we need them? And if malfunction occurs, they can cause autism. We don't want our president to have autism. And lastly, chloroplasts, they're only found in plant cells. They're not even everywhere, so how can we expect them to be loyal to everyone? And like mitochondria, they weren't even found in all cells. And we used to exist without them, so therefore we don't need them. We are the best candidate for presidency. We secrete proteins that function all over the body. We have multiple names: the Golgi apparatus, Golgi complex, Golgi body, or just the Golgi. We are foundation for the cellular infrastructure. We are the basis, the station for the cytoskeleton to come out of. And we are the shipping service of the cell. Without us, products wouldn't be able to move. They'd be stuck in their local environment, and information would not be spread within the cell and outside the cell. Without us, the cell would die. Vote for Golgi for president and receive free shipping for life. Thank you, everyone, and we expect your vote.